In this video, we will look more closely at the structure and function of DNA. This video will cover Chapter 5.2 through 5.5. So there are two big questions we're going to be addressing in this video. The first is, what is DNA? The second is, what does it do? DNA is a complex macromolecule. It is a type of nucleic acid that stores genetic information. DNA is the blueprint for who and what we are. DNA stands for deoxyribonucleic acid. This is a unique identifier, and no one in the world has the exact same DNA as you unless you have an identical twin. In Chapter 2, we learned that DNA is built of monomers called nucleotides. Each nucleotide is made of three parts. First is a phosphate group, second, a pentose sugar, third, a nitrogenous base. Nucleotides stack together and the phosphates bond to a sugar in a neighboring nucleotide. This creates a sugar-phosphate backbone with the bases sticking out to one side. DNA is double-stranded. Each nitrogenous base will pair with another base using complementary base pairing. This creates a ladder shape with two sugar-phosphate backbones on either side and the bases making the rungs in the middle of the ladder. The bases are connected by hydrogen bonds. In complementary base pairing, the base adenine always pairs with the base thymine, giving us A-T combinations. Guanine always pairs with the base cytosine, giving us C-G combinations. Because of complementary base pairing, we know that there are always an equal number of adenine and thymine molecules in a strand of DNA. There are also always an equal number of guanine and cytosine molecules. Let's practice with complementary base pairing. Below is a question. What is the complementary sequence of the DNA strand below? Take a minute and write down the complementary base sequence. We know that C's and G's always pair, and A's and T's always pair. The complementary base sequence for this DNA strand is G, T, A, C, T, A, G, G, C. Let's get back to DNA structure. DNA is in a ladder, but DNA is a double helix. This means the ladder must twist into a spring or corkscrew shape. Eukaryotic cells have so much DNA in each cell that the DNA must be packaged to allow it to fit in the nucleus. The DNA double helix is wrapped around proteins called histones. The bead-like clusters are then coiled into a thick thread called chromatin. When the chromatin is packaged into the nucleus of a cell, it looks like spaghetti. When cells are ready to divide or reproduce through mitosis, the DNA gets coiled up even tighter so that it is easier to move around. DNA structure was not discovered until the 1950s. In the 50s, James Watson and Francis Crick were the scientists responsible for putting together the pieces of the puzzle and figuring out the DNA double helix. Watson and Crick knew that DNA was built of nucleotides. They realized that those nucleotides bonded complementary to one another. They did not realize DNA was in a double helix until they gained access to X-ray chromatography pictures taken by Rosalind Franklin. They were given access to these by another colleague. When they looked at these X-ray chromatography pictures, they realized that the DNA molecules had to be coiled into a spring structure with the bases in the middle. However, Rosalind Franklin was not aware that they were using her data. When Watson and Crick published their paper, they did not give her credit in their initial publications. In 1962, Watson and Crick were awarded the Nobel Prize for their discovery. Rosalind Franklin did not receive the prize as she had passed away a few years earlier due to ovarian cancer. The rules are that the Nobel Prize can only be given to living recipients. It wasn't until many years later that Rosalind Franklin got her full credit for her work with the X-ray crystallography and the DNA structure. DNA is the universal genetic code. Every living organism, from the tiniest bacteria to the most complex organisms on Earth, have DNA that codes for their specific traits. However, the DNA sequence varies from species to species and organism to organism. These variations in the sequence itself are what code for who and what we are. Bacteria and other prokaryotic cells have a very small amount of DNA. Their chromosome is in a circle rather than a linear or string shape. Eukaryotes have much more DNA than bacteria do, and their DNA is packaged with histones and coiled into chromatin. Eukaryotes have many chromosomes, and the number of chromosomes varies depending on the species. For example, corn has 10 types of chromosomes, fruit flies only have 4 types of chromosomes, dogs and chickens each have 39 types of chromosomes, goldfish have 47 types, humans have 23 types of chromosomes. Individuals in each of these species inherit 1 copy of each chromosome from each parent. This means they have 2 copies of each chromosome. That means that total, they will have twice as many chromosomes as they have types. For example, humans have 23 types of chromosomes, but we have 46 chromosomes total. 1 of each type comes from each of our parents. If we look at the sequence of DNA, certain sections of DNA code for specific proteins. These are called genes. Genes are what actually code for our traits. Different variations of genes make different organisms. Let's hit a few vocab words. The genome of an organism is the full set of DNA an organism carries. This is all of the DNA inside their entire body. A gene is a segment of DNA that codes for a protein. A chromosome is a strand of DNA and the proteins that hold it all raveled up. An allele is the variation within a gene. So, if we're looking at a gene for hair color, the variations may be red, brown, blonde, and black. Those variations are the alleles. Here's another example of genes and alleles. Each gene is the instruction set for producing one particular protein. For this example, we see a gene that codes for flower petal color. Within a species, individuals sometimes have slightly different instruction sets for a given protein, and these instructions can result in a different version of the same trait. These alternate versions are called the alleles. For this example, we have 3 different alleles. One version, or one allele, produces red flowers, one produces white flowers, and the third produces yellow flowers. If we are looking at all the genes in an organism, we call this the genotype. The genotype is also what the DNA actually says, or what the sequence of DNA is. So the genotype is reading the genes. Phenotype is the physical manifestation of those instructions. This translates to what we see. So this can be said as the phenotype is the physical value or physical features that is coded for by the genotype. Let's practice for a minute. Which of the following is an example of 2 alleles of the same gene in humans? The answer is curly hair and straight hair. These are traits that are coded for by the same gene. Blue eyes and color blindness are 2 different traits. Long hair and short hair has more to do with environmental factors. Did you get a haircut today, or have you not cut your hair in months? Dark skin and freckles are 2 different traits as well. and therefore are coded for by two different genes. So remember, going from DNA to traits, our genes code for proteins. Proteins are the workers of the cell. They do different jobs. They can have structure and support. They can be enzymes that do chemical reactions. They can transport materials, or they can produce other molecules like pigments. So for example, when we have a gene that codes for eye color, we produce a protein that makes pigments that are laid down on the iris of our eye to give us different colors. If we put these terms in scale, we can see that genomes are the largest. It's all of the organism's DNA. The genome contains several chromosomes, which are strands of DNA. On each chromosome, we see hundreds of genes, segments of DNA that code for proteins. Each gene may have many alleles, which are different variations that produce slightly different versions of the proteins. Proteins are what produce our traits. It's important to note that not everything is genetically predetermined. We do see some environmental influences on our phenotypes. So, for example, if we think about human phenotypes, we have a particular baseline skin tone. Some are very pale, some are very dark. While this baseline is genetically coded for, many people can go out in the sun and get a tan. This changes our phenotype and changes our skin tone. Other people go out in the sun and develop freckles. We have already seen how many organisms have different numbers of chromosomes based on which species they are. It is also interesting to see that not all organisms contain the same amount of DNA in their genome. Humans actually have much less DNA than an onion, a salamander, or an amoeba. However, humans are more complex than these other organisms. So, DNA amounts does not correlate with complexity of an organism. It is also interesting to note that much of the DNA found in many organisms does not actually code for any proteins. For example, the proportion of DNA that codes for genes in humans is about 2%. Fruit flies is 19%, roundworms 25%, some plants 28%, and E. coli, a prokaryote, 90%. Many organisms have what we call junk DNA. This is DNA that is non-coding, so it does not code for a protein. Only a small fraction of the DNA in eukaryotic species code for genes. The function of this junk DNA is still a mystery. It may play a role in gene regulation, or it may just be unnecessary DNA that has accumulated over evolutionary time. It's interesting to note, though, that prokaryotes, like bacteria, have very little junk DNA. In eukaryotes, there are two types of non-coding DNA. The first is the junk DNA that we find in between the genes. This junk does not code for anything. We also see non-coding regions found within the genes. So there are areas in each gene that do not actually code for any part of the protein. If we look inside of a gene, the pieces of that gene that code for proteins are called exons. Exons will be cut and pasted together after the DNA is copied. A good way to remember this is that exons are expressed. They get made or translated into proteins. The non-coding regions inside of a gene are called introns. These regions will be cut out and discarded. Turning the code of DNA into a protein is actually a two-step process. The first step, transcription, reads the DNA and makes a messenger RNA. The messenger RNA is a copy of that code that can be moved out and then sent to the next step. Translation is when the mRNA is used to direct the production of a protein. This process requires ribosomes, the protein-making factories. Going from a gene to a protein is kind of like copying a cookie recipe from your grandma's cookbook. The cookbook is your original gene, the DNA. Okay? We have to copy that recipe down into something that we can take home. So if we copy the recipe onto a note card, we can take it back to our own kitchen. Transcription makes a copy of the DNA and copies it into a more mobile form, the mRNA. The mRNA molecule will then leave the nucleus and go to where the ribosomes are in the cytoplasm. With our recipe, we then have to gather our ingredients, and we can turn our words into an actual product, the cookies. During translation, the ribosome will read the mRNA molecule and translate that code into the actual product, the protein. In summary, all organisms have DNA, and DNA codes for proteins that make traits. We looked at DNA structure and how it is packaged inside of cells. We've looked at chromosomes and the varying numbers in different species. We also talked about the coding and non-coding parts of DNA. The next step is to look at gene expression. We will go in depth looking into transcription and translation of DNA.